Well, welcome everybody to today's exciting seminar of the Dana Farber Connect Science series. I'll remind you that you can access the schedule and future talks, as well as recording of prior talks on the website. And also please enter your questions in the Q&A tab at the bottom. It's my real pleasure to introduce today's speaker, Kevin Shannon, who did his undergraduate studies here in Massachusetts at Williams, and then he's been migrating slowly west ever since. He studied medicine at Cornell, and then pediatrics at University of Texas Southwestern in Dallas. He then joined the Navy, was a pediatrician at the Naval Hospital in Oakland for a few years before pursuing a pediatric hem-onc fellowship at UCSF, where he stayed on as faculty. He's done exceptionally well. He's now the Auerbach Distinguished Professor of Pediatrics. He has also had a number of institutional roles. He was for many years the director of the MSDP program at UCSF, and he now directs the UCSF Physician Scientist Scholar Program. He's been the interim chair of pediatrics twice, which I think means people must have liked him. He's received really a number of impressive prizes, including the Memorial Sloan Kettering Prize. He's a UCSF Faculty Basic Science Lecturer, a prestigious award from the UCSF. And his research interests have been focused on understanding the molecular pathogenesis of leukemia and of RAS pathway abnormalities. He's really made a number of seminal discoveries over the years. He was the first to describe that NF1 is a classical tumor suppressor by studying myeloid leukemias arising in patients with neurofibromatosis. He discovered that KRAS germline mutations cause Noonan syndrome, and he's really had a long-standing interest in studying the pathogenesis and the therapeutic targeting of aberrant RAS signaling in leukemias, as well as defining unexpected mechanisms of resistance. Kevin, it's a real pleasure. We look forward to your talk.
Thank you, Alejandro, for the very kind introduction. You've unfortunately sort of co-opted the first couple of slides I was going to show, but we'll do that anyway. So let me get started. I hope people can hear me. So I'll be talking about the palmitylation cycle as a therapeutic target in NRAS mutant cancers today. This is an area we've worked on for 7 or 8 years in the lab, and it only really, I thought, got ready for prime time to give us a seminar about in the last 6 months or so. So it's some new data, some newly published data, and I'd be welcome any feedback from people about how to give this talk better. I wanted to start today, though, by acknowledging an old friend and fellow pediatric hematologist oncologist, Alan D'Andrea, and to congratulate Alan on his election to the National Academy of Sciences for the seminal contributions that he's made in elucidating the Fanconi anemia pathway. It's a picture of Alan, one of my kind of favorites, because this is how he really looks in real life. Here's a picture of us at a AACR meeting in Beijing a few years ago, back in about 2008. One of the things that's inspiring about Alan is he sort of started with a human disease that affects the hematopoietic compartment, Fanconi anemia, and then has used that essentially bed-to-bedside approach to elucidating this incredibly important DNA repair pathway in normal biology and in cancer. So my pathway, I got hooked in sort of the same way as Alan did, but wound up by accident in a different pathway. And the pathway I wound up in is the RAS pathway. And as all of you know, this is a massive oversimplification, but RAS in cells is generally associated with membranes, particularly the plasma membrane. It's generally in an inactive GDP conformation. And in response to growth factors and other stimuli, a class of proteins called guanine nucleotide exchange factors become activated. They dissociate guanine nucleotide from RAS, and since there's a lot more GTP in cells than GDP, RAS then adopts this GTP-bound conformation, and that gamma phosphate stabilizes 2 domains of RAS called switch 1 and switch 2 that can then interact productively with effectors such as RAF and PI3 kinase. Now, like any good molecular switch, there has to be a way of switching it off when cells don't want to respond to a stimulus anymore. And RAS itself is an enzyme. It hydrolyzes GTP slowly. And a second class of proteins called GTPase activating proteins bind to RAS again, in these switch 1 and 2 domains, and accelerate the hydrolysis of GTP to GDP by thousands of fold. Now, this was all known back in the late 80s, early 90s. And what was also known was that when the common oncogenic RAS mutations at codons 12, 13, and 61 in RAS essentially lock the switch in the on position for 2 reasons. They have a variable effect on the intrinsic GTPase activity, but uniformly all of these mutant proteins are resistant to the activity of gaps. And that results in constitutively activated or elevated RAS GTP levels, and in many cases, hypersensitivity to different external stimuli, such as growth factors. Now, we got into the RAS field by accident, and Alejandro alluded to this. We were studying a rare cancer predisposition syndrome in children called neurofibromatosis type 1. And in NF1 patients, children, but remarkably not adults, are predisposed to an unusual type of myeloproliferative disorder called JMML. And we were able to show back in the early 90s that, in fact, this is, as Alejandro said, a tumor suppressor. And this was one of the informative experiments. This is a mother with NF1. This is her husband, who's homozygous for this particular microsatellite. You can see the normal tissues of their son with the normal allele from dad, the mutant allele from mom, and then loss of the normal NF1 allele. And the reason that this got us into the RAS pathway is that this tumor suppressor gene encodes one of the 2 major GTPase activating proteins, or gaps, for RAS in most tissues, including blood cells. And the young biochemist at Frank McCormick's company, Onyx Pharmaceuticals, we went on to sort of work out some of the biochemistry of this. So the NF1 protein is called neurofibromin, and in hematopoietic cells from mice and from patients that were lacking the NF1 gene, we were able to demonstrate that hypersensitivity to GM-CSF, the growth factor, which was a canonical feature of JMML cells, was in fact, could easily be reproduced by knocking out NF1 in cells, and that this led to increased activation of the downstream signaling pathways, particularly the RAS MEK-ERK or MAP kinase pathway. So this sort of serendipity, if you will, studying these unusual cancer predisposition syndromes sort of brought me into the RAS pathway, and I became sort of addicted to RAS in the way that cancer cells become addicted to oncogenic RAS, and have sort of never left thinking about RAS. And RAS is a very important oncogene, as you all know. It's particularly important in the KRAS in epithelial cancers. But in blood cancers, NRAS seems to be mutated much more frequently, about 2 to 3 times more frequently. Again, JMML, CMML, AML, the myeloid malignancies, having the highest incidence of RAS mutations. These data are from adults. If one looks at pediatric acute myeloid leukemia, it's something quite different and quite striking. And that is that NRAS is mutated in about 1/3 of pediatric AMLs, with these other signaling molecules, KIT, KRAS, PTPN11, and of course FLT3 being also mutated very, very commonly. By contrast, in pediatric AML, you see the most commonly, or one of the most commonly mutated adult genes, DNMT3A, is never mutated, and that P53 is very rarely mutated. So there really are rather dramatic differences between pediatric AML and adult AML, and this difference is also evident in young adults and adolescent patients. And I think this underlying difference in the genetic architecture suggests that our therapeutic approaches going down the road may need to be different in these different subsets of patients. So again, we became very interested in the role of these different RAS mutations in acute myeloid leukemia. And one of the first questions we asked some years ago is persistent oncogenic NRAS expression required for leukemia growth? And we did this a couple of ways. Ari Firestone in the lab developed some hairpins to knock down NRAS in pediatric in human AML cell lines. And in NRAS mutant cell lines, what you see is when you use a Ranilla hairpin that's a control, you see no change in the competitive fitness of these cells. But when you use either of two NRAS hairpins, you dramatically decrease the fitness and proliferation of NRAS mutant AML cells, but not KRAS FLT3 mutant AML cells. So that there is, even though we know from genomic studies that RAS mutations tend to be secondary or cooperating events in leukemia, they remain essential for proliferation of these AML cell lines in vivo. In addition, we, working with Kevin Heiges some years ago, generated acute myeloid leukemias characterized by NRAS G12D expression, where we injected these mice with the Maloney 4070 virus to induce multiple oncogenic integrations, and then turned on NRAS expression as a secondary event, and asked NRAS what it'd like to cooperate with to make AML. We've then used these AMLs and expanded them in vivo, transplanted them and used them for preclinical trials, but we can also use them to do biology. This is just an experiment Mike Burgess and Ari Firestone did, again, showing in these primary mouse AMLs that this hairpins knock down NRAS very efficiently. You can see that these cells repopulate very poorly in the bone marrow of mice. And you can probably see this best in terms of, here's the input in terms of the expression of the hairpins and the output in terms of the NRAS mutant hairpins versus the vanilla control. So very clearly, these cells are at a severe competitive disadvantage in vivo, and that of course translated to increased survival of these primary AMLs when we introduce these hairpins, with the most of these leukemias relapsing very late, and in every case we could find that the hairpin was no longer active and that NRAS was re-expressed. So this made us confident, at least, that it was worth pursuing trying to target oncogenic NRAS in leukemia. And obviously targeting RAS, oncogenic RAS and P53 are two of the great holy grails of cancer drug discovery. RAS is a very challenging target because the phosphate binding loop is structurally constrained, that the oncogenic mutations are genetically dominant, but result in lost enzymatic function. So they're sort of genetic dominance, but unlike BCR-ABL or mutant EGFR, they don't encode an hyperactive kinase. They actually encode a defective GTPase. So this repairing a broken enzyme becomes really problematic. RAS has extremely high affinity for GTP, and cells have lots of GTP. And RAS signaling is essential for normal development and cellular function. So a really challenging target from that point of view. Nevertheless, people have obviously been very interested in academics and in biopharma to try to figure out how to target RAS, and there are a number of different approaches to that. The first is, of course, to figure out a way to somehow directly inhibit the biochemical output of RAS oncoproteins, potentially by competing with GTP. This was thought to be undoable. Second, to do synthetic lethal screens to identify druggable targets that might be synthetic lethal with oncogenic RAS. One can think about inhibiting RAS effector pathways such as PI3 kinase or MEK or ERK or RAF. And one can think about blocking essential post-translational modifications. And just in a couple of words here, there's been a lot going on in this space. I think the most remarkable work has been by Keyvan Shokat, my colleague at UCSF, and a former MSTP student who's now at the Dana Farber named John Ostrom, who probably as an MSTP student was too dumb to realize that this was an undruggable protein. And working with Keyvan and Jim Wells, was able to develop the first G12C inhibitor. And as many of you know, these are now having some remarkable activity in patients, and it stimulated a lot of activity around can we also inhibit RAS proteins that don't have this covalent nucleophilic, where you don't have the covalent handle of the cysteine for nucleophilic attack. Whether that's going to be feasible or not, we don't know, but it's certainly exciting times. Synthetic lethality has been a very interesting area. Bill Hahn at the Dana Farber has been a leader in this area. I'll show some data later from David Sabatini's lab. The long and short of this is that it turns out that synthetic lethals probably do exist in RAS, but they may be very context specific. Finally, of the other three approaches, inhibiting RAS effector pathways, there's been very much activity in many labs, many pharmaceutical companies. Obviously, MEK inhibitors and BRAF inhibitors are approved for melanoma, but this has generally been disappointing to date in trying to target cancers with mutant RAS proteins. And so the last area is what I'm going to focus on today, which is blocking essential post-translational modifications of RAS proteins. So it turns out that RAS proteins are not highly homologous, but absolutely identical in this what's called this G domain, which is the N-terminal domain of the protein. And amino acids 10 to 17 bind the guanine nucleotide. The switch 1 domain that predominantly binds to RAF is amino acids 32 to 38, rarely to 40. And the switch 2 domain, which primarily binds PI3 kinase, but also GAPs and other proteins, is 59 to 67. And as I said, in all of the 4 RAS isoforms, KRAS4A, KRAS4B, HRAS, and NRAS, these are absolutely identical. So why do we have 4 of these proteins, and why are they different if the switch itself is the same? And the answer is that in the C-terminal end of the protein is a domain called the hypervariable domain. And in that hypervariable domain, RAS undergoes post-translational modifications that regulate its cellular sublocalization and its function. And going down to the B part of this, the initial modification that all RAS proteins undergo is farnesylation at this C-terminal cysteine, which is part of the CAAX or CAX box. This was shown in the 1980s to be absolutely required for RAS transformation. And this lipid modification is also essential for association with the plasma membrane and other endomembranes. As some of you know, the pharmaceutical industry in the 1980s and 90s developed potent and selective inhibitors of the farnesyltransferases. These were very good drugs in terms of their pharmacological properties. However, they failed in the clinic because when you inhibit farnesyltransferase, a second enzyme called geranyl transferase can take over in KRAS or NRAS mutant cells and perform an alternative lipid modification that, while not as efficient as farnesylation, restores the normal or oncogenic function to the protein. This is an important thing to consider because the failure of the farnesyltransferase inhibitors in the clinic cost billions of dollars in the pharmaceutical industry, and they made targeting RAS processing essentially radioactive for anybody's career. And so what that meant is it was up to us academics to sort of revitalize or restore interest in this area if that were going to happen. After the farnesylation reaction or this modification, the AAX protease called RCE1 clips off the AAX, the last 3 amino acids, and then a carboxymethyltransferase methylates the cysteine, and so you then increase the affinity of the RAS for membranes. However, the farnesylation is really the only essential step here. However, 3 of the RAS proteins undergo a second modification, and that modification is palmitylation, and that occurs on a different cysteine in the hypervariable domain. And the reason that this caught our attention is that one of the RAS isoforms, KRAS4B, which is the predominant isoform expressed in most cells, doesn't undergo this second site modification, but instead has this polylysine track in the hypervariable domain, and it directly associates with the plasma membrane through the farnesyl group and this polylysine track, this polybasic track, without undergoing palmitylation. And the reason I thought this was interesting when we first started thinking about it is from the point of view of a clinician. What we think about in cancer drugs is therapeutic index. How can we kill the bad cells and not hurt the good cells? And to me, this looked like something promising because essentially, if we were to do the thought experiment of inhibiting palmitylation in an NRAS mutant cancer cell that was dependent on NRAS, the normal cells in that patient would still be able to use KRAS4B to do the normal business that RAS normally does. And we knew from experiments that Kevin and others, Mariano Barbosa did, that in fact NRAS is not essential for mouse development, and so maybe a protein that we can inhibit reasonably hard in normal tissues and not see adverse effects. So this kind of got started down this pathway. So the palmitylation cycle for NRAS is fairly simple. It's on cysteine 181 after RAS is farnesylated, it traffics to the Golgi, and in the Golgi it undergoes palmitylation. And elegant work by Mark Phillips at NYU has shown that it's then trafficked to the membrane attached to a chaperone called BPS35. At the membrane, the palmitylated NRAS adheres or associates stably. It's then active in signal transduction. And this is a very active cycle in that palmityl transfer, in that serine hydrolases in the plasma membrane or in other endomembranes then remove the palmityl group and recycle the RAS back to the Golgi. And this is just another picture of this. You have the NRAS coming off the ribosome, it's farnesylated, traffics to the ER and Golgi, palmitylated, using the vesicular trafficking machinery, goes to the membrane where it's depalmitylated. There's membrane dissociation of this more weakly adherent protein. It gets trafficked back to the Golgi, and so the cycle proceeds. It's interesting from a biological point of view, and I think this cartoon shows this nicely, that this farnesylation reaction is irreversible, whereas the palmitylation reaction is highly reversible. And in fact, my collaborator, Ben Cravat, who will come up later in this talk in great detail, showed, looking at the entire cellular palmitome, that of all the many thousands of palmitylated proteins in cells, NRAS is one of the ones that is most dynamically palmitylated. It goes on and off the plasma membrane very rapidly, and again, this at least provides indirect evidence that this cycling of NRAS is very important in its normal function and also potentially in oncogenesis and in cell growth control. So the first set of experiments we did were fairly simple. They were done by Jin Su in the lab and published a long time ago, 2012. And what we did is just took normal hematopoietic cells, mouse fetal liver cells, and either expressed a GFP protein expressing wild type NRAS, NRAS G12D, NRAS G12D with a mutation that impaired but did not completely abrogate palmitylation, and a mutation and a protein with a second site mutation in the cysteine, the C181S, to abrogate palmitylation. And this is an old assay that it was the assay that was used first to show, in fact, that JMML cells are hypersensitive to GM-CSF. So JMML cells will track up here like these NRAS mutant cells. They form cytokine independent colonies and are hypersensitive to low dose of growth factors. You can see that this SSDD mutant is attenuated but still activated compared to wild type. But what caught our eye is that the C181S mutant was not only reduced to wild type, but was actually below wild type. It was actually impaired in terms of colony formation, both in terms of maximal growth and absolute numbers, suggesting that this protein had some dominant negative activity in the system, which admittedly is an overexpression system. And again, showing some of the biochemistry here, what you see is looking just at phosphorylated ERK downstream of activated RAS, wild type. The wild type protein has low basal levels and responds nicely to stimulation. The G12D version, the oncogenic version, has elevated basal levels, as one would expect, higher levels due to higher levels of RAS GTP, the active form of RAS, because of the broken switch I alluded to, again, is responsive to growth factors, not at the RAS GTP step, but at the ERK step. The SSD mutant, again, does load RAS GTP in response to growth factor. But interestingly, what we saw with the C181S second site mutant is lower expression of ERK and very little basal phosphorylation, elevated levels of RAS GTP, but lower levels of protein expression, suggesting that cells really didn't like this second site mutant protein and that it might have some dominant negative activity. And again, I got sort of excited by this, again, in an odd theoretical way, because if you think about it, the best cancer drug is not a cancer drug that turns a mutant protein into a dead protein. It's a cancer drug that turns a mutant protein into a protein that actually kills a cell or inhibits its growth. And we were beginning to see a little bit that this might be true here. So in collaboration with Herbert Waldman, he generously provided us with some inhibitors he had synthesized, which are called palmistatins. They're broad inhibitors of the serine hydrolases that remove the palmitoyl groups from proteins. And again, this is not anything to write home about, but just to show you, if we express NRAS G12D versus KRAS G12D and put this palmistatin inhibitor into a colony assay with either no GM-CSF or low doses of GM-CSF, what you can see is we could inhibit selectively and incompletely the growth of the NRAS mutant cells, but the KRAS mutant cells were completely refractory to this. And if we switch the domains of these proteins so that we took the hypervariable domain of KRAS and tacked it onto NRAS, or we took the hypervariable domain of NRAS and tacked it onto KRAS, we could reverse this drug dose response curve, showing that it was the hypervariable domain of NRAS, which contains the palmitylation site, that is required for sensitivity to this drug. We also did some immunofluorescence experiments showing that this particular protein, the treatment of cells expressing NRAS G12D or the KRAS molecule with the NRAS hypervariable domain mislocalized these cells, the protein away from the plasma membrane to the Golgi. So that was good. But of course, there's some problems here. And the biggest problem is that this is a sort of artificial overexpression system. And all of us who've been in RAS biology for long enough have the scars from overexpression studies from RAS. And so I began to think, could we do a proof of principle experiment to see if we could show that NRAS was actually essential for myeloid transformation? And this person photographed here is well known to many of you. This is Kevin Hygas, a long-term friend and collaborator from back in his time in Tyler Jack's lab as a postdoc, who's been a very generous collaborator over the years. And we've worked on various aspects of NRAS biology and oncogenicity in the hematopoietic compartment together. So we had shown in collaboration with Kevin, Ching, when she was in the lab, showed that if you take NRAS G12D, a knock-in conditional mutant allele that Kevin had generated, and you used MX1 CRE, which is a hematopoietic CRE, to turn this oncogene on, mice died with a fairly long latency with various hematologic disorders. And then Jin Zhu in the lab homozygosed this allele. And as I told you, NRAS is not essential for development. So one can make a perfectly normal mouse without any NRAS expression in embryonic life. And so we have this homozygous NRAS G12D allele. But importantly, now when we have 2 doses of the oncoprotein, we now get a very robust phenotype of a myeloproliferative disorder with these mice by 4 to 6 months of age, uniformly developing enlarged spleens with myeloid infiltration, high white blood cell counts, and a little bit of anemia from ineffective hematopoiesis. So this gave us a pretty good system to sort of ask the kind of genetic question that we were asked after. And so Kevin sent us his last vial of targeted ES cells, and we retargeted them and were able to recover them and send them back as well. And what Ari Firestone in the lab did is he retargeted the lock-stop-locks NRAS G12D mutant allele that Kevin had made by adding now, introducing a second site C181 substitution into the endogenous NRAS gene of mice. And the two bottom panels just show you that this allele works. This is probably the best or better confirmation. You can see looking at the GAT wild type going to, rather GGT wild type going to the GAT, G12 substitution, which is the most common oncogenic substitution in leukemia and other cancers. And also now back at codons 179 to 83, you can see that in the wild type, we have the G in the G12D, retains the G, but the G12D C889S, we've now made the G to C substitution, which encodes a serine to cysteine to serine. Importantly, when we look at expression of these proteins in primary cells, they're expressed at robust level, and using a G12D specific antibody, we can show that both a, if you will, wild type oncogenic protein and the second site mutant protein are both expressed very robustly. This is an experiment we did in collaboration with Jared Remsing in the Cravat lab, and this is all the work of Noemi Zambetti, who took over the project after Ari left the lab. And what I'm showing you here is, again, using the same G12D antibody with the input for NRAS. This is 17-ODYA labeling that shows palmitylation on NRAS. And to make a long story short, we only see palmitylation on the NRAS G12D with this G12D antibody, but not on the double mutant cells. These other lanes are hyaluronidase treatment to remove the palmityl and show that it's a specific palmitylation. So what happens to these mice? It was pretty dramatic. These are the double mutant G12D homozygous mice, and they uniformly succumb from this myeloproliferative disorder between, say, 6 and 9 months of life. By contrast, you can see that the mice expressing the G12D C181S protein are completely healthy and survive with normal blood cell counts. If we look at cell growth in culture, this is a cytokine assay in liquid culture growth. We can see the G12D cells are hyperproliferative in this assay, whereas the double mutant cells proliferate in the same way or grow in the same way as the wild type cells do. If we look at these cells biochemically, this is a phosphofax assay at different doses of stem cell factor stimulation. Stem cell factor actually, or c-KIT ligand, actually causes hyperactivation of ERK in the G12D cells, which you can see here and is graphed here. And you can see that in the wild type and C181S cells, the stimulation is equivalent and significantly lower. By contrast, when we look at phosphorylated A6 in response to stem cell factor c-KIT stimulation, what we see in these c-KIT selected cells is attenuation due to feedback in the G12D cells, but restoration of a normal response or normal activation in the wild type and the mutant cells. So again, these cells think they're wild type in terms of their cellular responses to cytokines and their biochemical responses to cytokines. So things got a little more complicated when we looked at the plasma membrane and looked at RAS itself. And so this is just a standard RAS GTP assays that all of you will be familiar with. And this is just whole cell lysates. This is the actual pull down with the RAS binding domain peptide that we use to pull down selectively active RAS GTP. And we look in the G12D mutant, and as expected, we see elevated basal levels, again, due to the decreased hydrolysis and response to cytokine growth factor. So this is GM-CSF stimulation. In the wild type, we see lower basals and response to the cytokine. In the C181S G12D, we also saw increased basals, which was not a particularly surprising, but what was surprising to me somewhat is that these cells respond by increasing RAS GTP levels in response to cytokine. Perhaps not as much as the G12D cells, but in repeated experiments, there's a definite responsiveness. And so we have a bit of a disconnect that we can activate RAS GTP in these cells. So this is just the total, and then this is the using the G12D antibody. We can activate RAS GTP in these cells, but they're not activating downstream effectors. And we think part of the answer for this is that RAS G12D, and I'll just focus you on this panel. If we look at the membrane-bound component of RAS, we can see that the G12D protein is primarily, I'm sorry, if we look at RAS G12D, it's primarily here in the membrane along with the wild type. Again, this is the G12D antibody, so we're not seeing the wild type, but the ratio of the RAS in the membrane with the G12D antibody is much higher. So you see here very little in the cytoplasm, a lot in the membrane. By contrast, more in the cytoplasm and less in the membrane. Now this may or may not represent, as I'll show you later, truly loss of RAS from the membrane, but the fact that during these preps, we may actually be losing some of the NRAS from the membrane because it's not stuck to it very efficiently. So the last part of the story that was intriguing to us was that we did do a cross where we crossed the G12D, if you will, wild type oncogenic allele to the G12D C181S allele. So now we had the G12D oncogene on both alleles and the C181S on one. And part of the rationale for this was, what if we had an inhibitor that could partially inhibit? This might be the sort of thing we might see. And what we got from this was a little bit surprising, is that we saw that the mice did develop myeloid diseases, leukemias. They developed sort of a very variable group of leukemias, and they developed them at a time where that was attenuated. It was much slower than the G12D over G12D. So they were attenuated, but they were still getting leukemia. These were quite aggressive leukemias. Some were transplantable, but they were very variable, and they implied to us that there must be some secondary events occurring. And the first place Noemi looked was at the RAS allele itself, and I'll just cut to the chase here since this work is published. Importantly, we can use this FRT scar from when we remove the hygromycin cassette in the targeting vector as a selective way of amplifying the mutant allele. And if we now look at these mice that develop leukemia, we can see, as we would expect, in the mutant allele, we have only the C, the mutant serine encoding substitution we made. But now in their bone marrow cells, what we see, in fact, is that there's been a revertant mutation. And that revertant mutation has essentially reversed the serine substitution that we made back to cysteine. And this just quantifies the fact that we're now reestablishing the, if you will, wild type cysteine sequence, showing that there's extremely strong selective pressure for these cells to somehow overcome this inability to palmitolate oncogenic NRAS in order to cause myeloid disease. So this is kind of the summary of that work. In the circumstance of NRAS G12D expression, the protein gets farnesylated, it gets palmitolated in the Golgi, it goes to the membrane, it has increased levels of NRAS GTP, it has low hydrolysis, so it stays in the GTP-bound conformation in response to growth factor signaling, hyperactivates RAF, MEK, and ERK signaling, as well as other pathways, and causes myeloid transformation. In the C181S, we think that the protein is tethered to the membrane. It's still GTP bound, but tethered to the membrane in an unstable way that results in much less efficient or no activation of RAF, MEK, and ERK, no transformation in vivo. And this protein falls off, goes back to the Golgi, where it can't be farnesylated, but sort of makes this endless cycle around, going back and forth from the plasma membrane. And we think that these data validate NRAS palmitylation as a potential therapeutic target, at least in NRAS mutant blood cancers. Now, what about targeting this on reaction? In other words, I show you these data and you say, good, let's go inhibit the enzyme that palmitoylates NRAS. And that's a really good idea, and it's something we're interested in and trying to pursue. However, just to give you some background about these palmitoyl transferases, which are not terribly well studied actually in biology, they do include a catalytic DHHE domain and a regulatory Golgi subunit. And these Golgi subunits seem to protect the DHHE domain from ubiquitin-mediated degradation. There are 23 different DHHE family members in humans and mice, one of which is known to palmitolate NRAS in a complex with another protein called Golgi 7, and that was shown back in 2005. However, genetics from mice and human, I'm sorry, genetic evidence, I flipped something in there by mistake. Genetic from mice and human evidence infers the existence of additional DHHEs that can palmitolate NRAS. And there are two strong lines of evidence for this. One is from Ribel-Wren's lab, where they knocked out DHHC9 in mice. And those mice actually developed a myeloproliferative disorder when that allele was mated to the Kevin's NRAS G12D allele using exactly the same kind of experimental design I showed you. So clearly, making the second site substitution that we made abrogated the disease, whereas knocking out DHHC9 did not. In addition, David Sabatini's dad did a lovely screen in AML cells for essential proteins for RAS transformation, or for RAS maintenance, if you will, in AML cell growth. And what they pulled out in their data was that Golgi 7 was selectively required for the growth, or was a dropout in the screen, but only in NRAS mutant cell lines. And so what this implies is that Golgi 7 may be required, but it may be able to form a complex with DHHEs in addition to DHHE9. And so obviously it's a bit daunting to try to figure out which of the other 23 can do this reaction, but we're taking a number of approaches to do that. But in the interim, we thought about, well, what about targeting the off reaction, the serine hydrolase enzymes that depalmitolate NRAS? And this is a paper that was published online last month, and I think is going to really get published like in print this week. The gentleman on the left is Ben Cravat, who's been a wonderful collaborator, who I called up about this and was totally game to work on this. He's one of the world's experts in serine hydrolases and chemical biology, and Micah Nefrakis at Abide Therapeutics, now Lindbeck, and a former postdoc with Ben, has been an incredible collaborator in terms of providing us with small molecules. And so I'll walk you through this collaborative project that we executed with Ben's lab. So the reason we kind of got interested in this, again, goes back a ways, and this was an experiment Ari Firestone did. And using the palmistatins are very unstable because of this beta-lactone bond. And so we really couldn't do cell growth assays, but Ari was able to figure out a window between 2 and 4 hours where he could study these molecules biochemically. And what Ari observed was the following. So if you look at NRAS mutant cell lines, and I'm just highlighting 2 here, and I'm just highlighting phosphorylated ERK. So these are 2 NRAS mutant cell lines, OCI ML3 and HL60. And what you see is in the first panel, those are cell lines that are treated with DMSO. In the second panel with a MEK inhibitor, PD901, which we would expect would, as a positive control for abrogating ERK phosphorylation. And then a palmistatin M at 2 different concentrations. And what you see is that the MEK inhibitor and palmistatin M greatly inhibited basal ERK phosphorylation in these 2 NRAS mutant AML cell lines, and in fact, in all NRAS mutant lines we've looked at. Now, by contrast, if you look at this KRAS mutant AML cell line NB4 or FLT3 mutant cell line MOLM14s rather, or KG1s, which have a c-KIT mutation, you see something very different, and that is only the MEK inhibitor inhibits you only see one hole here, and it's only the MEK inhibitor. Palmistatin is completely ineffective in these KRAS mutant lines. It's a little bit effective in the FLT3 mutant lines, perhaps due to the effects on NRAS signaling. It's completely ineffective in the c-KIT mutant line. And again, this was very exciting to me because this is that sort of kernel of therapeutic index that those of us who are physicians really think about when we think about any kind of cancer therapy, that somehow, some way, you might be able to shut off MAP kinase signaling, but only in the context of an NRAS mutation and not in the context of other mutations. And so, as I told you, palmistatin M and palmistatin B are not very usable either for cell line type experiments, for growth assays, or certainly in vivo. Micah's team at Abide and Lundbeck discovered a new class, a new chemotype of compounds that inhibited a range of different serine hydrolases. This was the compound ABD957 that we've worked with the most extensively. And using activity-based proteomic profiling, what you see is that this compound inhibits very selectively 3 enzymes in a family called ABHD17s, A, B, and C, as well as a couple of other serine hydrolases. And we were interested in these enzymes because of activity-based proteomic profiling experiments that the Conifer lab in Vancouver had done, and that the Cravad lab had done, suggesting that these ABHD17s were very important as NRAS palmityl serine hydrolases, or depalmitylases. And so the question was, can we build a system to really kind of interrogate this ex vivo? And what we decided to do is take OCI AML3 cells, which have an NRAS Q61 mutation, and express high levels of NRAS Q61, and as I've shown you before, dependent on this. And what Ari did is he knocked down this endogenous human NRAS and then expressed GFP fusions of NRAS G12D wild type or NRAS G12D with that KRAS hypervariable domain swapped in for the NRAS hypervariable domain. So we call the cells that express NRAS G12D ON cells, and we call the cells that express the NRAS hypervariable domain, the KRAS hypervariable domain, or the domain swap cells, ONK. And you can see at 21 kilodaltons here, there's really very low expression because of the very efficient knockdown that Ari was able to achieve. And so the Cravad lab, using ABD957, did a series of pulse chase experiments. And what you see is in the ONK cells, as you would expect, no palmitylation because we've removed that site. In the ON cells, we see in the DMSO treatment, and then after at the pulse, and then after the chase, you can see that the NRAS is becoming depalmitylated. And what you see here is PALMA-M and HDFP, another non-specific serine hydrolase inhibitor, inhibits that, if you will, off reaction, or that hydrolase reaction. ABD975 does it, but 2 inhibitors of other serine hydrolases, APT1 and APT2, that had previously been implicated as NRAS palmityl transferases, really have no effect. And this is just quantifying those data. And again, looking at these compounds, this is PALM-M, palmistatin M, and this is just the palmitylome of PALM-M that's being targeted, whereas there's very, very much greater restricted activity of the ABHD17 inhibitors. And what the Cravad lab noticed was, in fact, all of the targets of these ABHD17 inhibitors are actually present and active depalmitolysis at the plasma membrane. So they're localized at the plasma membrane in cells. And these protein targets then, presumably ABHE17 is acting upon them within the plasma membrane. So at a very localized context. And we think that this may explain in part the very exquisite sort of selectivity of these enzymes for just a few proteins in cells. This is an experiment again that the Cravat lab did in the ON cells that I showed you. And basically, I've told you palmistatin M relocalizes RAS to the Golgi, and you can see this probably best at this late time point. And this isn't nearly so evident with the ABHD, with the ABHE17 inhibitors, with the ABD957. There's some localization to the Golgi, but that's true of all RAS. And in general, if you image enough cells, what you see is that ABD957 is really no different in terms of its Golgi localization than DMSO treatment or the control, whereas PALM-M really massively mislocalizes these cells. And then looking more deeply into this phenomenon, the Cravat lab basically showed that looking again at the ONK cells, which have the KRAS hypervariable domain, you can see that treatment with the inhibitor has no effect on the subcellular localization of these cells. By contrast, you treat with the ABD 957 in the ON cells, and you see this mislocalization within the plasma membrane. So essentially, the NRAS is moving around within the plasma membrane from where it's normally localized to other locations more diffusely within the plasma membrane. And we've done some nice two-color analysis to sort of firm this up. If we look at the effects of ABD 957 and its ability to synergize with MEK inhibition. So this is in OCI AML3 cells versus NB4 cells. What we see is looking at phosphorylated ERK in the OCI AML3, the NRAS mutant cell lines, we see PALM-M markedly reduces phosphorylated ERK. ABD17 does to a lesser extent, but noticeably. PD901 is even more effective. And what we see in the NB4 and in all of the other non-NRAS mutant cell lines, we've seen no effect of PALM-M, no effect of ABD17, but effect of PD901. And this is quantified here. If we look at cell growth assays, it's only the NRAS mutant cell lines that are where their growth is partially inhibited by ABD957. And the KRAS mutant lines grow just fine. We noticed, and this has been true, that we only see partial inhibition with these inhibitors, and therefore realized we may need to do some synergy analysis and combine them with another less specific inhibitor. So this is looking at a Bliss synergy analysis in the ON, again, NRAS G12D mutant cells, and you see a high degree of synergy between this and the MEK inhibitor PD901, and no synergy in the ONK cells, and that's just quantified here. And in vivo, we've injected these cells, and again, to make a long story short, we do see synergistic inhibition of ERK phosphorylation and of cell growth between MEK inhibition and inhibition with and the 957 in vivo. So just to kind of wrap up, this is still a work in progress. Some of the questions we're thinking about with Ben's lab is how does mislocalization of NRAS within the plasma membrane impair NRAS signaling and transformation? We want to know if other serine hydrolase enzymes can depalmitolate NRAS. And we're interested in that because we see this partial effect of the ABHE17 inhibitors versus PALM-M. We want to know are the ABHE17 enzymes potential therapeutic targets in other NRAS mutant cancers. And I think the jury's kind of out on this. We know that hematopoietic cells are very cytokine and growth factor dependent. And pretty clearly, There's a lot going on at the plasma membrane with these inhibitors. Whether that will have the same effect in a thyroid cancer cell or a melanoma or in an NRAS mutant colon cancer cell is hard to know. We're planning to implant OCI-ON and ONK xenografts in primary AMLs and treat them with these 17 inhibitors and MEK inhibitors. We also, to test this kind of idea of chemical synthetic lethality in vivo. And we're also working, as I said, to identify the full complement of DHHE proteins that modify NRAS and AML cells. Just to wrap up, these are the acknowledgements for the people in my lab. Jin Zhu started this work, as I showed you in some of the cell lines. Ari was really critical in generating the mice and some of the reagents we used. Noemi Zambetti got a lot of this work across the goal line with the Cravat lab. Anaga was, and Amy and Marina were techs in the lab who helped the postdocs do this work. We're indebted to the Waldman lab and Frank Decker for the PALM-M, and to our collaborator, Amy Howell, for synthesizing it later on. To Kevin, who's been a great friend and collaborator over the years in the NRAS G12D space. To Jared Rimsberg and Radu Sachi in the Cravat lab, who did the heavy lifting on a lot of the data I showed you at the end of the talk, and to Micah and Ben. This has really been a fun collaboration for us, and I've learned a fair amount, or a little bit anyway, about chemical biology. So I'll leave that slide up as instructed by Claudia and be happy to take any questions.
Thanks very much, Kevin. That was terrific, really exciting. I'll remind the audience to please enter your questions in the Q&A tab. The first question is from Vijay Sankaran. How does specificity of ABD957 serine hydrolase inhibitor vary across different cell types? Could there be off-target impacts on other palmitylation targets, such as Rho GTPases in other cell types?
So the answer is for sure, yes. I would say, Vijay, though, that the Cravat lab has done very extensive analysis of the palmitolone in various cell types, and at least in terms of proteins that are dynamically palmitylated, NRAS and actually HRAS do come up as being dynamically palmitylated, but I'd have to look more carefully. But I think the other GTPases, for whatever reason, don't seem to be as dynamically palmitylated as RAS. Now, the fact that we could abrogate NRAS palmitylation in the whole animal genetically, I think does say that if you were to do this therapeutically in cells, you could get away with it for NRAS. Whether we could get away with it for N and HRAS, it's a little hard to know. Whether these inhibitors act on HRAS, we're not really sure. So there is a possibility of sort of non-hematopoietic toxicity, I would suppose, but I think it's reassuring that you can generate the C181S second site mutant, and that these mice are okay. As you know, MX1 Cre doesn't act in some tissues, so I guess the jury is kind of still out on that. It's a good question.
And I've got a question. I think I just missed this, but it seems a little, on the most simplistic level, counterintuitive that either blocking palmitylation or blocking RAS in the palmitoylated state would both block signaling. Do you think this suggests that RAS is signaling during the switch or during, as it moves?
This is a really good question, and we've talked about this a lot. I mean, it sounds a little bit counterintuitive that both the on and the off reaction would have the same biological effect. There's some very nice work by the Rox lab that was published in Cell a few years ago that actually showed that, in fact, that this is a very highly regulated cycle. And if you inhibit the off reaction or the serine hydrolase reaction, NRAS tends to sort of wander around to get mislocalized in cells and sort of loosely adheres to endomembranes. And I think part of the explanation for this is that the endomembrane compartment in cells is so large that in order for it to efficiently move between the Golgi and the plasma membrane, it really has to be a highly regulated process. And if you disrupt that regulation on either side, either at the front end or the back end, you do wind up inhibiting the efficiency of the signaling reaction.
And one more question is from Wei Tong. Mark Phillips found that NRAS is largely cytosolic and only some in the membrane due to retention by the VPS35 chaperone, in contrast to other RAS proteins. I wonder if AML cell lines might be different.
Yeah, Mark is a wonderful scientist, and his VPS35 work is just phenomenal. So I think the whole question of endomembrane signaling and Golgi signaling by RAS is a really interesting one, and may contribute to the phenotypes. I think having these kinds of chemical inhibitors that Ben's lab has developed will really help to elucidate some of those questions. I do think that in our hands, in hematopoietic cells, and these are all myeloid lineage cells, most of the NRAS in cells is at the plasma membrane. And we do know that hematopoietic cells are very cytokine responsive and cytokine dependent for making cell fate decisions. But as I alluded to, I do wonder as to how specific this dependency might be for hematopoietic cells and other cell types where NRAS signaling may be more endomembrane driven, could have much less of a dependency on this sort of ABHD17 plasma membrane mechanism. So it's a really good question. I don't think we really know the answer. I think the whole area, I think this kind of work both on the cancer side and on the normal side does sort of raise this whole sort of specter of trying to understand better as cancer scientists the whole area of subcellular localization, vesicular trafficking, and how that actually contributes to or impairs oncogenic activity in cells.
Well, thanks very much. We're over time. That was really stimulating. I'm excited to see where this goes. And thanks to everybody for attending.
Thanks for having me. Thanks for hosting me, Alejandro.